Good afternoon, everyone. My name is Ryan Bannon. I'm the Director of Partnerships and Events with Life Sciences BC, and I'd like to welcome you to today's event. Just a reminder that all attendees have their video off and are on mute. However, we do encourage you to be very interactive during the afternoon by posting questions in the Q&A section. We also have the ability to upvote on the questions so that you're if you want to hear a question answered, it goes to the top of the list. And we also have a chat function as well. And so without further ado, it gives me a great pleasure to introduce Wendy Herbert, who's the President and CEO of Life Sciences BC. Over to you, Wendy.
Thanks, Ryan. Hello, everybody. Thank you very much for joining us at our McCarthy Spotlight Speaking Series. We always look forward to these great events where we bring together leaders under a specific topic and hear about latest research developments, latest advancements in science, and have an opportunity of thinking about what the future is going to look like. As we begin, I'd like to acknowledge that I'm joining this event from within the ancestral, traditional, and unceded territory of the Coast Salish peoples, including the territories of the Musqueam, Squamish, and Tsleil-Waututh First Nations. I'd also like to acknowledge that others are joining us from other traditional and unceded territories across this beautiful province, country, and maybe even around the world. Welcome to today's webinar, and thank you for joining us. We're very excited to be here and to host this event virtually. Over the past 2 years, Life Sciences BC and our community have fortunately embraced virtual events. We're grateful that we have been able to continue to bring together the community, both in BC and abroad and across the country, to host webinars and have discussions and just generally bring our ecosystem together. We're excited about today's event, the McCarthy Spotlight Speaking Series, a very long-running series with Life Sciences BC, so thank you, McCarthy's. Today, we are going to be focusing on oncology. Now, for some of you that may be new to Life Sciences BC, a little bit about us. We're a not-for-profit organization. Our members consist of academia, research institutions, centers of excellence, funding agencies, and emerging small and medium-sized businesses in the areas of digital health, medtech, therapeutics, and many of our members are companies that support the broad life sciences ecosystem, all under the vision of creating a thriving ecosystem in British Columbia. We're funded through membership and the generous contribution of our sponsors and partners. So this event was made possible by McCarthy Tétrault, and specifically David Frost, the partner at McCarthy, who has a long-standing relationship with Life Sciences BC. We want to thank you very much for your investment of Life Sciences BC and the life sciences ecosystem broadly. It is invaluable, and we really appreciate it. At each event, we'd like to introduce our newest members. So today, I would like to welcome Boasis Therapeutics, Inc., Nanovation Therapeutics, and Xenia Technologies, Inc. Welcome to the Life Sciences BC family. We're always welcoming new members, so please reach out to any of us at Life Sciences BC or go to our website, lifesciencesbc.ca, for more information. And now I would like to introduce David Frost, partner at McCarthy Tétrault. David is a partner in the Vancouver office and co-chair of its National Life Sciences Group. He practices in the areas of corporate commercial law, securities, and corporate finance with a focus on financing and merger and acquisition transactions involving biotech companies. David regularly advises private and public issuers and dealers on cross-border US and domestic public and private offerings of equity and debt, including initial IPOs, bought deal financings, and secondary offerings. He also advises public companies on general security law matters, including continuous disclosure obligations and other regulatory compliance issues. David regularly acts for Canadian, US, and international acquirers and targets on domestic and cross-border merger and acquisition transactions and takeover bids. He also advises Canadian and US venture capital investors in connection with the structuring and implementation of domestic and cross-border investments. David has been recognized by Lexpert as a leading lawyer in health sciences and corporate mid-market work, and a financial and corporate law star by LMG Life Sciences. David's a passionate supporter and leader within the life sciences sector, and we're so thrilled to have you with us again and your ongoing support. I think there's very few organizations within our biotech sector that you haven't had some form of involvement with over your career. So thank you, David, and over to you.
Oh, thanks very much, Wendy, for the kind introduction, and welcome everybody to today's McCarthy Spotlight Speaking Series. McCarthy's been a proud sponsor of this series for many years, and our goal when we put this together was to put the appropriate spotlight on certain sectors of our industry, of our space, make sure that we can share knowledge and inform people about what the community is doing, because there are so many people in the community who are doing fabulous things. And today's topic on oncology, we have a terrific roster of people that I'm so proud to introduce. And so without further ado, I'll introduce our first speaker, Dr. Francois Bernard. Francois is Vice President Research at BC Cancer, a distinguished scientist at the BC Cancer Research Institute, and professor in the Department of Radiology at the University of British Columbia. He holds the BC Leadership Chair in Functional Cancer Imaging. As a clinician scientist, his research interests are in positron emission tomography, nuclear medicine, cancer imaging, and radioligand therapy. His team developed several new radiopharmaceuticals targeting tumor receptors, notably peptides and small molecules ligands. He initiated the program that developed cyclotron production of 99-MTC, which completed clinical trials at multiple sites in Canada. He has established extensive multidisciplinary collaborations, and he and his colleagues were awarded the 2015 Brockhouse Canada Prize for Interdisciplinary Research in Science and Engineering by NSERC. Over to you, Dr. Bernard.
Thank you, David. I have only a short amount of time, so 8 minutes, and I'll try not to delay all the others to present a little bit of the activities at in technology commercialization at BC Cancer. BC Cancer has been involved in commercializations for over 3 decades. And the typical approach when we have to decide about whether to pursue an invention disclosure and pursue commercialization activities, and the key criteria is, for us, this is a tool to advance clinical care. The public sector cannot on its own turn a discovery into a commercial, a product that will have a tangible impact on cancer patients unless it goes through a commercialization process. And the key criteria for us is really to advance the standard of clinical care. So typically, we will first assess the researcher commitments and the passion, of course, to engage as inventors and founders. There's an in-house assessment by an office at the Provincial Health Services Authority called the Technology Development Office that looks at the technology, the intellectual property portfolio, competitive advantage relative to other technologies, and the value proposition as well. And there's multiple approaches to fund the technology. Non-dilutive grants are usually quite useful to advance the technology to a better state of readiness. And then obviously there's usually company creation or seed funding or a licensing agreement made with an established company. And the TDO accompanies the researchers in the process of commercializing their discoveries. So it's been a long journey, and like all public institutions, BC Cancer and PHSA have learned from prior history and past history about things to be very careful with when negotiating licensing agreements. One key issue that keeps coming back is a definition of licensing improvements, because the public organizations or the academic institutions want to maintain academic freedom. We also need to make sure that other researchers who may not be involved in that inventions are not impeded in their ability to function and continue their research or clinical work. And it's sometimes a challenge to align BC Cancer and partner needs. And the state of the art in terms of defining what is an improvement over an invention have led to some practice changes over time, notably to limit the definition of improvements to the researchers are actually involved in the invention, aligning it with the scope of the patent. For example, a typical clause nowadays would be to make sure that the improvements are defined by the valid claims of the patent or within the scope of the valid claims. and also provide a timeline as well, so that researchers, once the company is up and running with the technology, that it doesn't bind or commit researchers or the institution to unreasonable terms. Conflict of interest is also an important part. We're a public organization and publicly funded, so we do need to take into account having a fair and transparent process to manage the conflicts of interest. So BC Cancer typically has a research budget that will oscillate between 65 to 100 millions, depending on whether it's a year with big infrastructure investments or a year where, like last year, where COVID happened and reduced some grant funding opportunities. We always have a large number of advantageous disclosures. Provisional patent filing applications range usually under 15 to 20 per year. But obviously the national patent applications follow that trend as well. And there's always, every year, we have licensing transactions with the private sector, either through the creation of startup companies. For example, we've had 2 to 3, Cuprous, Artemis in 2019, Alpha9 and Innovakine in 2020, and a startup company called Vita as well last year. And this brings back revenues, both in terms of reimbursements for the legal fees, but also bring back revenues from the sale of equities when the shares become more liquid, as well as bringing gross revenues every year to our ecosystem at BC Cancer and PHSA. And it can reach, like last year was a good year with almost $3 million in revenues from commercialization activities. So the timelines can be sometimes quite long. As an example, I've got a couple of case studies or companies that were created as startup companies. In 1999, the first invention disclosure was for a drug formulation by Dr. Bally and Lawrence Mayer. And the company was incorporated in 2001. And that eventually led to a new drug for acute myeloid leukemia that was approved by the FDA in 2017. And this is an example of a success story. And that led eventually for Celator to be acquired by Jazz Pharmaceuticals for $1.5 billion following a successful phase 3 study. Another example is ESA Pharma, which is an active member of our ecosystem. And that's from an invention disclosure by Dr. Marianne Sedar and Raymond Anderson at UBC. The company became public in 2015 and is now in phase 1-2 studies currently with their drug candidates. We have another very different approach. Is Cytopex Bioinformatics is a company that was initially created by Dr. Brinkman based on software that had been developed in the open source model. So he created a service company based on know-how, and the company is a company specializing on novel methods for analyzing flow cytometry data. And this was acquired by Insightful Science, which is a company that has a large portfolio of science-based software. And the company was acquired in May 2020. And the products obviously now have a funding partner to continue developing its assets. Another example is my colleague, Dr. Brad Nelson, who co-founded in 2019 a company called Innovakine Therapeutics. This is a company that developed new technologies for receptor engineering for CAR-T, for therapeutic T cells that are reinfused in patients. And the company has a rich IP portfolio and is now in preclinical studies to develop second generation or next generation cell-based therapies for cancer treatments. Another example is a company I'm involved in. I co-founded this company in 2019 with Dr. David Perrin and Dr. Koscianlin. This is a company developing novel radiopharmaceuticals for cancer imaging and therapy. The company secured $11 million US in Series A financing in 2021. And is currently undergoing, is at the clinical stage now with clinical studies. So there's a large number of companies in our ecosystem that you will recognize, either companies that were founded by BC Cancer affiliated researchers or were startup companies, including all the way back to stem cell technologies, which grew out of the BC Cancer Research Center. And was led by, has been founded by Dr. Alan Eaves and is now Canada's largest biotechnology companies. And obviously, we have a lot of companies you'll recognize in our ecosystems that have ties to BC Cancer. Thank you very much.
Great. Thanks very much, Dr. Bénard, for that overview of sort of the ecosystem and the community. Next up is Jim Pankovich. Jim is the Executive Vice President, Clinical Development at Bold Therapeutics. He leads a team of dedicated research professionals developing Bold 100, a novel ruthenium-based small molecule for the treatment of patients with gastrointestinal cancers. In his career, Jim has been responsible for the development of investigational agents for the diagnosis and treatment of oncology, infectious disease, and inflammatory conditions. Mr. Pankovich has more than 30 years of research and development experience working for small to mid-sized biopharmaceutical companies in Canada and the United States, as well as 6 years as the Chief Scientific Officer for the CIHR Canadian HIV Trials Network. He has an MSc in Experimental Medicine from the University of Alberta and an MBA from Queen's University. Over to you, Jim.
Well, thank you very much. And thank you very much for the opportunity to speak at this speaker series. What I will do today is introduce you to both the company Bold Therapeutics as well as Bold 100, which is our novel cancer therapeutic in development for the treatment of patients with GI cancer. I'll start with here's a slide actually from the BC Cancer Agency, some data over the last 20 years showing the improvement in a lot of the 5-year survivals for many of the standard cancer indications. And you'll see here that over that period of time, there's been quite a considerable improvement in the treatment and diagnosis of patients with cancer. Having said that, though, you'll also see in the bottom left-hand corner of this slide, there's actually still a lot more work that we can do to help patients be able to deal with the diagnosis and treatment in cancer. And so what we've done at Bold Therapeutics is really looked at this problem is that current oncology treatments for advanced cancers are often ineffective or result in high relapse and refractory rates. And we think we've got a potential with Bold 100 to significantly improve some of these outcomes in patients by helping overcome some of this drug resistance. You'll see a little bit of a corporate snapshot here. It's been a busy couple of years since the founding of the company in 2018, and the last couple of years, 2020 and 2021 was really getting up and running with a lot of our preclinical work, as well as the initiation of a second phase 1B clinical trial. You'll also see from this that 2022 is going to be an extraordinarily busy year with manufacturing, some additional clinical work that will be going on, as well as looking at our preclinical work and deciding on which additional indications we'll want to move forward with. The Bold 100 pipeline is really quite full. Our initial indications here are with Bold 100 in combination with FOLFOX. And you'll see there we're targeting GI cancers, and that includes advanced gastric cancer, pancreatic cancer, colon cancer, and bile duct cancer. But we've also got, through our preclinical data, and I'll show you some of that data in a moment, we think we've got additional opportunities in other indications, including esophageal cancer, multiple myeloma, and sarcoma. We're also in the process of looking for additional products, what we call Bold 200, to acquire to have our team further develop both preclinically and take them into the clinic. So Bold 100 really has some unique advantages. And again, I'll show you a brief snapshot of some of the data in a moment. But it really combines well with chemotherapies, targeted agents, and other immunomodulatory agents. So it works very well with much of the current standard of care therapy that is utilized in oncology. It also has can work in a wide range of solid tumors as well as liquid tumors. It can be effective against both sensitive cell lines as well as cell lines that have become resistant. It addresses a lot of the unmet medical needs that I show you on that first slide. There's still definitely a much greater need for development of new and novel therapeutics to be able to help some of those patients that are not getting the responses that they deserve. The product itself will have an orphan drug designation. We've been issued both pancreatic and gastric cancer orphan drug designations. And we'll be, as we get our clinical data through this year, we'll be looking at various breakthrough designations, which can become very important from a regulatory and a commercialization perspective. The product itself, we think, has blockbuster potential, particularly with the number of indications that we believe it can work in, and we've got robust intellectual property worldwide. A little bit of mechanism of action, and really important for a product like Bold 100 is it's got a unique mechanism of action. You'll see the molecule there in the middle. It's a unique, it's a ruthenium-based molecule that when administered to patients really has an effect on a couple of different really important pathways in oncology. On the left, you'll look at the unfolded protein response. And this becomes very important for cells that are under stress, in particular cancer cells that are under metabolic stress because of rapid growing, or maybe under stress because of treatment with cancers, with cancer therapies. So with that, for the cell to be able to survive in that scenario and to be able, it actually upregulates the unfolded protein response that allows that cell to maintain and become more immortal. And what Bold 100 does is it actually downregulates that unfolded protein response that is induced by the stress and then makes that cell much more susceptible to cell death. At the same time, Bold 100 has the ability to actually induce reactive oxygen species. Again, with the induction of these reactive oxygen species, it leads to single and double stranded breaks in the DNA, alters the cell cycle, leading to cell cycle arrest, and then again, leading it towards cell death or apoptosis. So through one or both of these mechanisms, depending on which one of the cell types we're talking about, Bold 100 can have an effect of helping kill the cancer cell and help the cancer cell overcome some of the resistance to some of the therapeutic agents. Here again, just a bit of a snapshot of the data. On the left-hand side, we think the value of a product like Bold 100 is to really move it through in combination with other chemotherapy and targeted therapy agents. And the data on the left is really just a snapshot showing that we've used this Bold 100 in combination with many standard of cares in many different cell types. And you'll see there almost all of the ones that are shown here either result in either synergistic or additive cell killing. Importantly, again, on the right is really looking at how we can combine this together, and this using an in vivo model where we're looking at an N97 gastric cancer model. This model is typically considered to be sensitive to cisplatin therapy, which is the orange line there. In this model, Bold 100 by itself as a monotherapy has little effect, and that's the blue line. But when you combine both the cisplatin and the Bold 100 together, which is the green line, you'll see that the median tumor burden is really decreased in this very aggressive gastric cancer model. As well, we talked a little bit earlier about overcoming some of the resistance, and resistance refractory cancer is a very difficult to treat cancers. So using a multiple myeloma model, on the left here, you'll see this is combining Bold 100 with two common protease inhibitors. The first is bortezomib, and the second is carfilzomib, which are both typically used as proteasome inhibitors to treat multiple myeloma. In the first set of slides there, or first panel, you'll actually see the treatment with Bold 100 with one of these proteasome inhibitors, and you'll see in both of those cases there's an additive effect. Interestingly, though, is once we actually start to develop some of the resistant cell types, and in this case, the second set of panels is really around bortezomib resistance. In a resistant cell line where we add Bold together with that proteasome inhibitor, we're seeing strong synergy. Again, overcoming some of the resistance that's been generated to the bortezomib alone. And again, in the last panel there, you'll see that even when combined with another proteasome inhibitor, we help the Bold 100 combined with that proteasome inhibitor helps it to overcome the resistance that's been generated. Again, on the right-hand side, it's just the same, looking at this with some of the immunomodulators that are utilized. And again, looking at the cell resistance, the resistant cell lines to both the proteasome inhibitor in the top and the immunomodulators in the bottom, you'll see here that with the treatment of BOLD 100, the resistant cell lines are actually much more sensitive than to BOLD 100 than they are to the other agents. Again, looking at overcoming some of the resistance, and this is a specific model that we're looking at in terms of pancreatic cancer. On the left, this is a pancreatic cancer model that's a very resistant to gemcitabine therapy, and you can see that from both gemcitabine and BOLD both having modest effects. But it's only when you combine them both together where you're seeing about a 300% improvement in the median survival in these animals when treated with BOLD and gemcitabine together. Interestingly, on the right-hand side, this is a live tissue sensitivity assay that we're working together with Moffitt Cancer Center in Florida. And we're looking at taking tissue samples right directly from patients and then using it to determine which type of therapy they would move forward with. So on the top panel, this is really a gemcitabine paclitaxel panel. And in the bottom, it's a FOLFIRINOX panel that's utilized for this live tissue. And what you'll see here is the green bars in every case. When we combine the standard of care therapy, whether it's GemPax or FOLFIRINOX with BOLD 100, you're getting a decreased viability, meaning that there's an increased cell killing in every one of these tissues tested to date. This is our current clinical trial. We're moving quickly through the dose escalation phase and into the cohort expansion. And you'll see here that we're combining FOLFOX with BOLD 100. FOLFOX is a standard of care for most of these GI indications. We're going through our dose escalation, and very shortly we'll be moving into the cohort expansion. This is a trial right now that we're doing primarily in Canada, but I'll show in a moment we're going to be expanding it internationally. Currently, we're doing the dose escalation portion of the trial with 6 Canadian clinical trial sites, and we're moving to add 2 sites in the US and 5 sites in South Korea as we move into the next phase of the study. Just to give you a snapshot of some of the preliminary data from the first 13 patients. Again, keeping in mind the phase 1B is really a safety and tolerability in combination with FOLFOX. We're seeing that it can be safely combined with no new grade 3 or 4 toxicities. We also can see that patients can be treated for extended treatment cycles, with 1 patient being treated actually now and beyond 15 cycles. And we are starting, even in these early studies, we're starting to get indications of clinically meaningful improvement in progression-free survival over FOLFOX alone, with 1 patient having experienced a 48% reduction in the tumor target lesion. We do have regulatory approval to move forward in the United States and South Korea as we move forward into the next phase. We're a data-driven company, and here you'll see that right now we've got our current clinical trial sites, the 6 sites that we're working with in Canada, as well as in the US. We'll be working with Moffitt Cancer Center and UCLA. We do have a regional pharmaceutical partnership with Hana Pharm in South Korea, and with that, we've also got 5 additional clinical trial sites we're working with. And you'll see on the right, we do all of our scientific work through research collaborations with collaborators around the world, although we do have a specific focus on wanting to work with Canadian researchers that have specific expertise in some of the work that we're doing. We have a dedicated group of professionals, some of which are on this slide here, but many more that are helping in support of the work that we're doing, both clinically in the clinical development as well as our non-clinical work. And just lastly, will be just our contact information. If you're interested in hearing more about our product or our company, there's some contact information there. Thank you.
Well, that's great. Thanks, Jim, for letting us know about BOLD 100, sharing some information about it, and letting us know what BOLD Therapeutics is up to. So our next speaker is Dr. Stephanie Willard. Dr. Willard, a full professor in biomedical engineering, holds a Canada Research Chair in biomedical engineering at the University of Victoria, where she has dual appointments in the Department of Mechanical Engineering and the Division of Medical Sciences. She also holds an appointment with the School of Biomedical Engineering at the University of British Columbia. She recently founded the startup company Axolotl Biosciences that sells high-quality bioinks for bioprinting human tissue models. She is an active member of the steering committee of the BC Regenerative Medicine Initiative and the Stem Cell Network. She also serves as a staff scientist at Creative Destruction Lab. She served as the acting director of the Center for Biomedical Research and the Biomedical Engineering undergraduate program at UVic from 2018 to 2021, and as the president of the Canadian Materials Society from 2018 to 2019. Over to you, Dr. Willard.
Thanks so much. Yeah, that was a really long introduction. So I'm actually going to talk mostly today about my startup company, Axolotl Biosciences, which uses bioprinting. And I'm going to talk about some of our really exciting work modeling cancer. And it looks like I'm going to need to flip this to be the right view. Display settings. Okay. So our goal at Axolotl is to develop superior quality reagents for 3D printing human tissue models, making research and drug discovery easier than ever. And today I'll be talking about how bioprinting can be used to enhance some of the preclinical tools for cancer drug development. So 3D bioprinting, similar to traditional 3D printing, where you have a computer file containing your design, but instead of using a plastic to secrete the shape that you're desiring, it uses specialized bioinks that are designed to keep the cells inside alive and develop the structure of the tissue you're hoping to replicate. And some of the really interesting things with bioprinting is that it really allows you to recapitulate some of the complex biology you find in the human body, including that associated with cancer biology. And what our company has really chosen to focus on, some of you may be familiar with one of our collaborators on the Willard lab side, Aspect Biosystems, who makes high-end bioprinters like you can see here. Our company instead focuses on the reagents and making unique inks that really support human cell models, and in particular, personalized models of disease. Just briefly, you can see the largest demand for bioinks currently is with academic researchers, but I think as you'll see with the example I'm going to give today, it's going to become increasingly relevant to clinicians, especially as we make this process of bioprinting simplified and easy enough to print these human tissues on demand, which you can then use to screen potential drugs against, including those for cancer. Our company currently has a product on the market called Tissue Print, which you can see in the middle, which is a picture of our product. It's been published in 6 different peer-reviewed journal publications, but essentially our bioink is really good at keeping human cells alive, and in particular delicate human stem cells, as well as other human primary cells, which I'll be talking about in a little bit. And if you want to read more, I just suggest going to my Google Scholar profile. And I will point out that the initial paper, when we published, one of the initial formulations of tissue print was selected as the method of the year for ACS Biomaterial Science and Engineering. And outside of our wheelhouse, although today's the Oncology series, some of you may have seen me present before, our ink was initially developed with the idea of printing lots of different types of neural tissue. And in fact, I'll be talking about our complex human tissue model of glioblastoma that we've printed. But it also works really well for printing all sorts of different human cells, ranging from cardiac cells to testy organoids, all the way to bone and umbilical vein endothelial cells. And so if you're looking to make human tissue models of cancers or even of healthy tissue, I definitely suggest reaching out to me and our team at Axolotl. So the model I'll be talking about today is using 3D bioprinting to mimic human glioblastoma. Obviously, tumor cells move in a 3D environment. A lot of work's been done on looking at how to model these 3D tumor microenvironments. Glioblastoma is a horrible disease for anyone who has known someone who's had it. It's the most common malignant form of brain cancer originating from the astrocytes. And pretty much the standard of care hasn't changed in 20 years, which might be one of the common themes you hear today. So there's a real need to find better ways of treating these types of cancer, because in the case of glioblastoma, it almost universally recurs and often is almost always fatal. So this work that was initially led by Chris Lee, who was a undergraduate in the Willard lab, and then he did his master's with me in Håkon Nygaard over at University of British Columbia. He used the Aspect bioprinting system along with tissue print in order to print these human glioblastoma cells into rings. And interestingly, when you culture these cells in 3D, they act a lot like tumors do in the brain, and they actually formed as you can see here, they actually spontaneously formed tumor spheroids inside of our 3D printed constructs. And this is much more relevant if you're trying to screen a drug against a model. And probably those of you who are familiar with this field, you've seen this where people have used major gel to induce these spheroid formations, because it's really easy to kill cells that are grown in 2D, but actually breaking apart a spheroid and replicating that biology is much more difficult. And that's one of the reasons why bioprinting is so powerful. So after we published that initial paper in Materials Today Chemistry, Zonula, a Montreal-based company focused on cancer therapeutics, reached out to us with this compound you can see here, which is an NCAD-adherent antagonist that they had previously shown could break up breast cancer tumors, and they wanted to test it in our model. And so Imka Schmitz, who was another amazing graduate student we had, she actually used bioprinting. And this is why it's such a powerful technology. And she actually printed these cancer cells alongside healthy human astrocytes in a co-culture model. And she did something really interesting, which was she allowed these cultures to grow for 16 days before we treated them with drugs. Because normally when you're treating a cancer in vivo, you've detected it because it's formed a tumor, not that, and then that's what you'd actually have to break up. So this is actually a really interesting biological model for a couple of reasons. One, it's a completely human system. Two, you can actually test if your drug is going to selectively kill off the cancer cells as opposed to the healthy tissue. And in this case, she actually used labeled cancer cells. So you can actually see at day 16, the tumors across all the different conditions. And then she treated them with different concentrations of drugs, as you can see here, with the higher concentrations of the Zonula drug being able to kill and break apart these tumors while sparing the human astrocytes. And she went ahead and quantified this. And if you want to read more, this was published in 2020 in Biochemical and Biophysical Research Communications. And I won't go into it, but there was a lot of detail showing why you actually needed the bioprinting to get the proper mix of cells and to get the architecture and things like that for this co-culture model. And more recently, we've received some patient-derived glioblastoma cells that we're hoping to do similar work with from David Nathanson at the University of California, LA. And this is where I think 3D bioprinting really has a lot of power, is the ability to actually take your patient cells and culture them in these humanized tissue models and screen drugs specifically on the variety of, in this case, glioblastoma that you're taking from the patient to see if it actually responds and gets broken up or killed by the drug you're trying to test. And this is just our team at Axolotl. I'm the CEO and co-founder, along with Dr. Laura De La Vega and Lila Abelseth, who helps make the tissue print formulation. We currently have 6 staff who work on production and things like that. And we're always happy to talk more if you're interested in testing out our inks for a variety of things. Before I encourage you to follow us on social media, the other thing I just wanted to briefly point out about our models is they are fully human systems. So compared to when you have a human tumor in an immune compromised mouse, we're actually hoping to work with some of the BC cancer researchers, and we've been in talks with them to actually do some of the CAR-T cell therapies and evaluate them in our humanized system, because you would actually have spike in all the human immune components and things like CAR-T therapies, and have a fully humanized system for evaluating these potential cancer treatments in an in vitro setting, which is going to more accurately mimic what you'd find in a human body compared to having a human tumor in a mouse system. And so with that, being conscious of time, I'll turn it back over to David. And again, thank you to Life Sciences BC for letting me present. And again, follow us on social media and feel free to reach out to me if you have any questions about our technology.
Perfect. Thanks very much, Dr. Willard, and thanks very much for letting us know what Axolotl is up to. Our next speaker is Farzian Aminuddin. Farzian is the Director of Business Development at Virogin Biotech, where he identifies partnering opportunities, manages external collaborations, and is involved in the company's product development strategy. Prior to joining Virogin, he was part of the business development team at the Center for Drug Research and Development, now known as MRNA Bioinnovations. As a business professional, Farzian has worked in Vancouver and Toronto, developing academic industry partnerships to translate early stage life sciences technologies. As a researcher, his focus in the genomics led to several publications in the field of respiratory disease. Farzian is an MBA graduate with a biotech background and a focus on business innovation. Over to you, Farzian.
Thank you, David. Let me just quickly share my screen here. All right, well, thank you, David, and special thanks to McCarthy and for sponsoring this event, and for the Life Science BC for giving me the opportunity to present, in which I will spend my precious 8 minutes here to highlight how Virogin Biotech is tackling cancer with oncolytic virus therapy. But before I get into the details and the uniqueness of our platform, what I want to do today for a few minutes is to really explain the value of oncolytic viruses so that everyone's on the same page right off the bat. So firstly, the cool thing about oncolytic viruses is their ability to preferentially kill off tumor cells without affecting the healthy cells. But we all know that cancer is a complicated disease. So the best way to kill this metaphorical bird is with multiple stones. And that is why at Virogin, we love to play with oncolytic viruses, because on top of the destruction of the infected tumor cell, these viruses will also trigger a local immune storm that will destruct the tumor microenvironment as well. Not only that, but viruses also replicate and can spread to infect more tumor cells. And when these tumors lyse, they release tumor antigens that gets picked up by the immune cells that will lead to a more systemic immune response. And on top of that, oncolytic viruses can be engineered to express payloads like cytokines, chemokines, and antibodies to really enhance the therapeutic effect. And all this can be done with an oncolytic virus. So at Virogin, we're taking these viruses and we're stepping up its game, because even though everyone recognizes the potential, the clinical benefit so far has been disappointing, to say the least. So how is Virogin solving this? Firstly, we're using the oncolytic herpes virus, HSV1, to express IL-12 and IL-15 with its receptor alpha subunit. What sets us apart from the payloads others are using is that IL-12 and IL-15 work together with great synergy to activate not just the adaptive, but also the innate anti-tumor immunity. Now, the second feature we have incorporated in the HSV, the herpes virus, is what we call the transcription and translational dual regulation, or TTDR, which is the use of tumor-specific promoters and microRNA regulation to boost the oncolytic effect in a very tumor-specific manner, rather than just attenuating the virus, which many groups do to prevent the unintended toxicities. We have data to demonstrate that we are not compromising safety at all while enhancing the virus backbone to make it more potent. So let's take a quick look at what we're seeing so far with our lead program in order to first validate the use of the IL-12 and IL-15 cytokine combination. So our first product candidate is VG161. This is an attenuated version of HSV, but expressing these cytokines along with a PD-L1 blocking peptide. We're on the tail end of finishing up our phase 1 study, looking at patients with advanced solid tumors. And so far, the data is quite promising. On top of having a good safety profile, we're seeing tumor reductions in both the injected and non-injected tumor sites, which supports our product's ability to really trigger a systemic anti-tumor activity. Now, what's most promising are the initial signs of clinical benefit observed in some patients. And keep in mind, this is all still done in a monotherapy setting. So we're now advancing to evaluate VG161 in combination with other agents, such as checkpoint inhibitors, while we expect to see better, better clinical activity. So now I want to introduce you to VG201, which is our first oncolytic virus expressing the cytokine combination, IL-12 and IL-15, but also incorporating our TTDR feature, which is that unique backbone and that has shown to be much more potent. We have data demonstrating that this version is more strong, potent, and as safe as an attenuated version. But one thing I want to highlight here is evidence showing a strong and durable systemic anti-tumor effect. In an immunecompetent tumor model, what we've done is we've injected the oncolytic virus into a tumor on one side of the mice and observed the tumor size and tumor growth on both the injected tumor and a distant non-injected tumor. And what you can appreciate here is complete tumor regression in most mice over 7 weeks in both the injected and non-injected sites. So what indicates is it's indicating a strong immune-mediated killing of tumor cells. Now, we've got more exciting stuff and plans ahead with this type of backbone in VG201 in particular. So I encourage you guys to stay tuned on over the next coming months. Now, I want to shift gears a bit here and spend the last few minutes talking about quite a newsworthy item. As you know, we are focused on realizing the full potential of oncolytic viral therapy. And we are quite excited about our oncolytic virus platform, but we're always looking for the next best thing. So what are we cooking up in the lab these days? We are now looking to developing our very own mRNA tumor vaccine platform, which will be designed specifically to be used in combination with our oncolytic virus to really enhance the anti-tumor effect. Now, let me explain a bit more on how this would work. Whenever you have an oncolytic virus introduced in your body, we cannot get away from the antiviral immunity our body creates as a protective measure. So to overcome this challenge and to improve the lifespan of the virus to do its thing, what we're proposing is a strategy in which the mRNA vaccine primes the body against the tumor before boosting the effect with the oncolytic virus. And what you get at the end of the day is the immediate dominance of the anti-tumor immunity overshadowing the antiviral activity being generated. So why are we using mRNA vaccines? Well, unless you've been hiding under the rock these days, I don't need to have to tell you too much about the stability and the effectiveness of LNP-delivered mRNAs. So this is all still in discovery stage, but we are quite excited so far on how we could truly improve the way that we aim to eliminate cancer. So just like to spend my last minute here to showcase a bit the brains behind our platforms. When we have got an experienced leadership that's been there, done that. We've got a great clinical stellar advisory board. A special shout out to Dr. Brad Nelson from BC Cancer. We've recently closed a financing round, which then now totals our private financing of 300 million USD raised to date. This has allowed us to scale up our activities both in Vancouver and Shanghai sites, create meaningful partnerships to really penetrate the largest markets like China and the US, and transform from being an SME into a vertically integrated biofarm with our plans to really to manufacture our own GMP facility. So I encourage you to keep us on our radar as we work on our vision to create impactful drugs for patients of the highest medical need. So thank you very much for your time, and back to you, David.
Great, thanks very much for that, Farzian, and sharing that information with us and giving us a little insight into what you're up to. Thanks for that. So our next speaker is Dr. David Mulder. So Dr. Mulder is an assay development scientist at Canexia Health, working to develop clinically validated tests that prove equity of access in cancer diagnostics and monitoring. He trained in the fields of genetics and genomics at SFU with Fiona Brinkman, at BCCDC with Sui-Han Go, at McMaster University with Brian Coombs, and developed a hybrid capture T-cell immunoprofiling method with Trevor Pugh at UHM. He joined Canexia Health in 2018 and has contributed to the development of the Fusion's assay for partner agnostic detection of solid tumor clinically actionable structural rearrangements. That's quite a mouthful. And the liquid biopsy follow-it V5 assay for detection of actionable variants in tumor-derived circulating cell-free DNA. When not in the lab, he pursues hobbies in the wilderness and in the kitchen. Over to you, David.
Thank you. Thanks for inviting me to present. Hopefully everyone can hear me and see my slides. So my name is David Mulder, and I'm a scientist at Canexia Health. Today I'm going to talk to you about how Canexia Health is connecting Canadian cancer patients with precision medicine. So we are Canexia Health, a clinical cancer diagnostics company based in Vancouver, BC, founded in 2012 as Contextual Genomics. We are a multidisciplinary team of clinical, laboratory, and computational professionals. We develop laboratory-developed tests for oncogenic DNA variant identification in partner labs. Our goal is to connect cancer patients with personalized therapy with speed and confidence with technology that powers the entire clinical testing process. Our laboratory developed tests are validated and performed in partnered labs. So that's on the first left side of the bottom screen. Then our informatics platform is used by the partner lab to identify and report alterations and to generate a report for transmission to the clinical professionals. So first, I'd like to talk about Follow It, our liquid biopsy assay. So in a liquid biopsy assay, we start with blood instead of your typical tumor tissue. At any given time in our body, cells are dividing and dying. When cells die, DNA is released into the blood as circulating cell-free DNA. So we can isolate this cell-free DNA from only 2 vials of blood collected at a local Life Labs or any other lab collection site. And then the assay performs multiplex amplification of this cell-free DNA, and our platform is used to identify DNA alterations, including single nucleotide variants, insertions, deletions, copy number alterations. So as you might know, COVID-19 has disrupted cancer testing worldwide. In Canada, more than 100,000 surgeries have been delayed, with almost half of those being cancer surgeries. It has been reported that even a single month delay in cancer treatment can raise the risk of death by almost 10%. So in 2020, we launched Project ACT, Access to Cancer Testing and Treatment in Response to COVID-19. Through ACT, we have committed to providing 2,000 cell-free DNA tests to cancer patients in Canada. And through ACT, we are seeking to encourage provincial coverage for cell-free DNA testing and to establish local cell-free DNA testing infrastructure across Canada. So to date, Project ACT has made a difference in nearly 2,500 patients in Canada, and reports have been issued with identified alterations in over 1,200 cases. Over 400 oncologists at over 100 institutions have ordered tests for patients with metastatic breast, colorectal, and lung cancer. And this project is providing rapid clinical liquid biopsy cancer testing access to any Canadian with advanced metastatic disease to guide precision medicine therapy with a 10-day turnaround and requiring only 2 vials of blood. Next, I wanted to bring up the Findit and Fusion solid tumor assays. So unlike liquid biopsy assay, this is these are solid tumor assays. For these assays, we isolate RNA or DNA from surgically resected tumor tissue and perform the lab processes and variant calling as before. The assay performs multiplex amplification as before, and our platform is used to identify alterations, including single nucleotide variants, insertions, deletions, copy number alterations, and for this case, microsatellite instability, as well as gene fusions. So why gene fusions? Well, gene fusions drive many types of cancers. They are caused by rearrangements within or between chromosomes. Oncogenic gene fusions involving kinases are typically actionable with small molecule therapeutics. And in this figure here on the right, we can see that many different types of cancer along the bottom axis and fusion driver genes along the left axis. So I want to point out, highlighted in red, the thyroid cancer, THCA, which has heavy involvement of kinase gene fusions, including RET, BRAF, NTRAX, and ALK. So fusions of such genes, such as the RET gene, can be targeted with novel small molecule inhibitors, such as LOXO292, a recently approved drug. So understanding the type of alteration involved in thyroid cancer is important for treatment selection, such as LOXO292. A bit of background about thyroid cancer. It's the most common endocrine cancer in Canada, with 8,600 new cases and 230 deaths in 2020 alone. Papillary thyroid cancer is the most common type and is driven predominantly by single nucleotide variants in the BRAF gene and fusions involving the RET gene. It has been found that incidence and prevalence of these alterations varies considerably between international cohorts due to environmental and social cultural factors, and no large analysis of driver alterations in papillary thyroid cancer has been done on a Canadian cohort. So we wanted to know who would benefit from RET fusion drugs in the BC population of thyroid cancer patients. So to do this, we performed retrospective testing of 95 papillary thyroid cancer specimens collected from Vancouver General Hospital between 2017 and 2020. We performed histopathological testing, including immunohistochemistry and fluorescence in situ hybridization to identify ALK gene fusions, RET gene fusions, and BRAF gene single nucleotide variants. And we also performed NGS testing with the fusions assay and Findit assay to identify single nucleotide variants, insertions, deletions, copy number alterations, microsatellite instability, and gene fusions. So as a result, we identified oncogenic drivers in 98% of the samples, so that's great. 8 samples were found to be fusion positive, 77 were found to have BRAF single nucleotide variant positive, were found to be single nucleotide variant positive for BRAF using IHC. Interestingly, 8 additional samples were found to have BRAF variants when we added NGS testing, and 2 samples were negative by all tests. So overall, only 2 samples were negative when we used testing involving histopathology and NGS testing. So between the use of these 3 assays, Follow-It, Findit, and Fusions, I hope you can see that NGS testing adds value in cancer care. Identification of oncogenic drivers with these assays enables access by patients to small molecule precision therapy. Project ACT has allowed access to NGS testing for more than 2,000 Canadians with metastatic disease. The liquid biopsy platform has reduced morbidity of testing and does not require a hospital visit. Additionally, the Follow-It assay permits monitoring for emergence of drug resistance variants over time. And then next, testing of papillary thyroid cancer patients with the Findit and Fusion assay showed that addition of NGS testing allowed detection of alterations targeted by precision therapy in 8 cases and detection of rare gene fusions in 3 cases. So these would not have otherwise been picked up. And additionally, NGS testing was robust with all archival samples passing all QC tests, allowing for 3 additional cases to be identified, as 3 of them did not pass standard pathology QC for the histopathological testing. And so in summary, NGS testing enables precision therapy, which enables better outcomes for patients. And with that, I'd like to thank my colleagues at Canexia, as well as our partners and the audience and the organizers. Thank you.
Thanks very much, David, for sharing that information about Canexia. So our final speaker today is Dario Passerini. Dario's professional experience includes 15 years managing biotech sales and marketing teams with a focus on North America, Europe, Asia, and the Middle East. His education includes a BA in language translation, as well as a marketing management certificate from the Sauder School of Business at UBC. Over to you, Dario.
Thank you very much. Let me just share my screen. Thank you very much for, hang on a second. My apologies. I'm having some technical difficulties here. Could you please let me know if you're able to see my screen?
Not yet, Dario. We saw it briefly.
Okay, 1 second. Here we go.
We can see it and then just presentation mode.
My apologies, everyone. Here we go. Perfect. I believe you are able to see my screen now, right?
It's absolutely perfect now.
All right, sorry about that once more. So my name is Dario Passerini, and I work for Signalcam Biotech. SignalChem Life Sciences Corporation is a clinical stage company developing novel targeted therapies for oncology. Our unique business model has been built upon important pillars: the drug discovery development, so SignalChem Life Sciences, the bioreagents and research services business, the SignalChem Biotech, the in vitro diagnostics development, SignalChem Dietech, and the plant biosynthesis system, or SignalChem Pantech. And for the sake of this presentation, I will focus today on SignalChem Biotech, the bioreagent and research services side of our business, which is where I work. So SignalChem Biotech is founded in 2004. We're located in Richmond, BC, and we're a global leader in protein engineering, manufacturing, purification, and assay development. What's the SignalChem advantage? All of the departments that are involved in the life cycle of a protein production, so from cloning to quality testing of the products to approval of the manufacturing process and quality testing, are all housed under one roof. And also, our products are all packed in-house and shipments are tracked by a logistics team to ensure the product is delivered in the hands of our customers. This shortens turnaround time and makes fast and effective resolution to any technical shipment development issues that we may have. We develop highly active proteins with a team of scientists with extensive experiences in not only novel enzymes, but also developing assay methods to test the functionality of these proteins. We have proven this through our ever expanding product portfolio and successfully delivering custom proteins to our customers. Throughout our whole protein production process, every single department involved is managed by PhD scientists, and we like to make sure that we have the best and most active proteins for our customers and partners. We do have a very large kinase portfolio. It is the largest kinase portfolio in the world, and we offer a total of over 5,000 different products. Products appear in over 1,200 publications in high-impact journals. And just to give you an overview of the different product lines that we offer, but for the sake of this presentation, I will keep it brief and quickly talk about just a couple of them. So kinases, SignalChem's reputable specialty. Kinases catalyze the phosphorylation of substrate by transfer of phosphate groups. Deregulation of kinases could lead to major human pathological conditions, such as cancer, cardiovascular disease, neurodegenerative diseases, and metabolic disorders. Therapeutic inhibition of kinases is a huge area of research in cancer therapy. Our active kinases product line is one of the most comprehensive in the world in terms of the coverage and has been utilized by many pharmaceutical companies in drug discovery programs. Mutant kinases. Genetic alterations such as mutations in protein kinases alter their biological function and dysregulate the cellular pathway they are involved in. So hence mutations lead to pathological conditions ranging from cancer, inflammatory disorders, cardiovascular diseases, to neurodegeneration. So it is a very hot topic as well in research, and we offer more than 300 clinically relevant active mutant kinases. Phosphatases, it's the opposite process of kinase. Phosphatase is an enzyme that removes a phosphate group from a protein. Protein phosphatases constitute a large family of enzymes, which are crucial modulators of cellular phosphorylation events. Dysregulation of these phosphatases is associated with autoimmune diseases, cancers, and cardiovascular diseases. So they're also a very attractive therapeutic target. Ubiquitin enzymes, the ubiquitin enzyme system can be used as an alternative to kinase inhibition in drug discovery process and has recently become a hot area for research. So in our cells, proteins are continuously made. Some of them may not be properly folded. So to degrade these proteins and make room for new proteins, ubiquitin enzymes mark or tag them for degradation by proteasome. There are three types of ubiquitin enzymes which work in coherence in vivo. All of the three categories, E1, E2, and E3, are listed on our website, signalchem.com, and you can take a look over there if you'd like. Epigenetic enzymes, they're emerging with huge interest as druggable targets as well. They catalyze these modifications and therefore are responsible for coordinating gene expression. Epigenetic targets are some of the most promising classes of druggable targets in oncology research and include histone deacetylases, acetyltransferases, methyltransferases, and arginine deaminases. Products have a broad range of applications and are used in screening, Western blots, crystallography, binding studies, as well as custom application. And also, being any protein, we have extensive experience with range of expression systems to tackle almost any custom protein project that you may have. So bacteria, yeast, insects, plant and mammalian cells as well. Lastly, we provide profiling services where a customer can take advantage of panels of over 600 kinases or epigenetic enzymes or ubiquitins. And overall, this is just a very quick overview of what SignalCam Biotech is capable of. And I'd like to invite you to, again, to check our website at signalcam.com. At SignalCam, we like to say that we manufacture the puzzle pieces that fit your research. And so we'd love to help you with any of your research needs. Thank you again.
Thank you very much. Okay, so this wraps up. I know we're a little bit over time, but this now wraps up our first McCarthy Speaking Series of 2022. I want to thank David Frost again and the McCarthy Tétrault team for your continued support of Life Sciences BC in this long-running speaking series. Personally, I found this an excellent session today. Dr. Bernard's overview of how BC Cancer works in commercialization, and then from all the companies that were speaking. So thanks very much for your excellent presentations. We know you have a lot of demands on your time, so we really appreciate you taking the time to share your insights and stories with us today. We always end each event with an overview of events that are coming up that we want to draw your attention to. So in a few weeks, we have our annual Access to Innovation Conference on February 16th and 17th. We have a very impressive line of speakers that have been committed, and we continue to add speakers every day. So please check that out. You can reach out to any of us or find information about it on our website. In March, we have a new event this year that we are piloting, and it's Invest in the West. So we will have early stage companies from across Western Canada and the territories pitching to investors and to the community. In addition, our 10 companies that are graduating from our investor readiness program will be presenting as well. Axolotl is one of our graduate companies from last year. And then actually this date is incorrect. Our Vancouver Island series has moved to the first week of April, and more information will come on that. So again, thank you very much. We really appreciate you joining us today. As always, please give us any feedback that you have regarding our events. And if you have any ideas of topics that you'd like to hear about, we're always looking for fresh ideas about what our community would like to learn about. So thank you to our sponsors. Your support of us is invaluable, and we very much appreciate working with you and all the work you do to advance the ecosystem of British Columbia. Thanks, everyone, and have a good rest of your week.